Namaste to all. Welcome to the next session on molecular biology and rDNA technology. In this session, we are going to see about spontaneous and induced mutation. In our previous session, we have seen about different types of mutagens and different types of mutation. In this, we are going to see that how the spontaneous and induced mutation causes the damage in our DNA. So mutation in types of cell, if we see, as we have seen in our previous, it can be divided as somatic or vegetative cell and germinal cell, that is lining of gonads. If it is taking place in somatic cell or vegetative cell, then it is called as, example for this is skin cancer. If it is taking place in gonads, then it can cause the damage in the sperm or in the ovum. So if it is taking place in the somatic cell, it is non-transferable to offspring. But if it is occurring in the germinal cell, it is getting transferred during the zygote formation. So based on its process, if you see, it is divided into two. So one is spontaneous mutation and other is induced mutation. Spontaneous mutation means without our intervention. But induced mutation means it is by our involvement. That is, by extrinsic factors. It can be like drugs, it can be radiation, it can be various other factors. Spontaneous mutation. Those that arise in the absence of mutagens are called as spontaneous mutation. They occur spontaneously. Genetics recognized them naturally. They are natural genetic variations that are observed in population. Copies are required, then mutagens has to be induced. Spontaneous mutation rates varies, that is, large target tends to mutate more frequently. The rate of mutation here is 2 to the power of 12 into 10 to the power of 6 mutation per gamete per gene. It leads to number of hereditary diseases. So occurs due to various sources, that is during replication, that is you can see during replication, consider this is our sequence, that is TT, CGA, CGA, AGC. So the other strand is also having AA, GC, TG, C, TT, CG, which are complementary. Now, this is DNA before replication. So during the replication, what happens is the strands get separated and it has to be synthesized or elongated. So consider one of the things, like one of the strand in which it is getting separated, if the synthesis taking place and in place of C, if T is added, then that will lead for the mutation here. So this is a replication error which has occurred due to the lack of or missing of the exonuclease activity during the proofreading. So what happens is it will lead to the formation of one with normal daughter strand and the other one which is having the defect, that is the TG pairing has taken place and it leads for the some kind of point mutation or mismatched pair has taken place. Spontaneous lesion. So this also causes the transposable elements. These are the various types in this. So the replication error and spontaneous lesions, if you see, that leads to the base substitution and frame shift mutation. Replication error only we see, then it also leads to deletion, occurs in the absence of mutagenic treatment. So replication error and mismatched pair. So how it occurs? So let me see about the transmission mutation. So if you see the transition mutation, the T is becoming as C. So the pyrimidine to pyrimidine change during the mutation. A is becoming as G, that is purine to purine mutation. So change. So this is called as transition mutation. Transversion mutation, if you see, this has a conversion of T becoming as or pairing with A as well as with you can say like the other one, that is C. So that is pyrimidine to purine and purine to pyrimidine. So both the changes can take place here. So you can see here in this particular system or in the particular diagram, that is thymine has to pair with adenine basically. But in this particular scenario, what happens is basically they are going to pair with another base, um, that is pyrimidine, which is there. They will also pair with guanine here. Okay, so thymine to adenine, thymine to guanine pairing, which can occur, that is pyrimidine to purine, and cytosine to guanine and adenine pairing. In the same manner, the purine to pyrimidine, if you see, adenine actually pairs with basically the thymine, but here they can pair with cytosine also. So and guanine can pair with cytosine and thymine, so both the things they can pair with. So this is what is actually the transversion mutation is. So frame shift mutation. So in the frame shift mutation, as we have seen earlier also in the frame shift mutation, so one base, if it is getting deleted or added, then the frame is read in a different manner. So if you see here in this strand, GATCTCA, G is there, then GCTAA is there. So what happens here is in the DNA, if the G is removed, then what happens is this is the deletion of the base. Due to this, the reading changes here. So when the bases has to join, or when the daughter strand has to join here during the transcription, what happens is it is joining as CUA, GAG, then U. Then it is having, instead of CGAUU, what happens is it is getting changed and the mutation is taking place. So the protein what is formed here, it will have the deformity in this. So like normal has to be having leucine, glutamine, serine, asparagine. But now what happens is because of this mutation, it has formed as leucine. So you can see here, this is how the normal is present here. But now since it is converting, so this is going to become as leucine, glutamine, serine, isoleucine. So you can see here, this is how the effect of the deletion will take place. The whole thing normal, from the normal protein, it has got a deformed protein, or a change in the protein has taken place. So the deletion and duplication. So based on the deletion and duplication, so if A, B, C, D, E, F is there, if A, C, D are getting deleted, in that if C gets deleted, it becomes as A, B, D, E, F. It will give another kind of protein. If duplication is going to occur, then what happens is, you can see here. So when the duplication occurs of D, then it becomes as A, B, C, D, D, E, F. So this is how the duplication is occurring, and it leads for the various changes in the protein. So spontaneous lesion. So if you see the spontaneous lesion, that the types of spontaneous lesion, that is depurinization, deamination, oxidative damage. So in this depurinization occurs due to the disruption or breakage of the glycosidic bond between nucleotide base and deoxyribose. So what happens here is it releases the base of adenine or guanine and causes a purinic site in the deoxyribose. Due to this, it leads to mutation by incorporating the correct base during replication. So you can see here, since the base has got removed here in this is called as a purinic site. So during the replication process, since this base is not there, there can be more problem while forming the other daughter strand. So deamination. So if you see the deamination process, the process involves the removal of amino group. So from the molecule which causes the base change, and you can see here, because in the cytosine the sugar is attached, cytosine is there. So the amine group is NH2. Because of the deamination process, it has become the carboxylic group, and it has changed from cytosine to uracil. And due to this uracil, what happens when it is getting repaired? So when the repair has to take place during the replication, it pairs basically with adenine. So the pairing actually is considered, but during the process, if it is getting deaminated, it can become uracil, but it has, in the DNA, uracil should not be there. So the changes has to take place. So the causes, another thing is in the transcription process, if it is occurring. So in the DNA strand, if it is becoming deaminated from cytosine to uracil, and then it has paired with the adenine. So now what happens is adenine. So what happens is the causes GC pair to become as AT pair in the later stage. And during the transcription, what happens is the product what we have to get here, there can be a minor change, because in the place of guanine and cytosine pair, the thymine and adenine is there. So when the protein formation takes place, it again becomes as uracil. So when it, or adenine. So what happens is when it becomes like this, the changes are again going to take place in the protein also. So oxidative damage. So oxidative damage, if you see the damages, the oxidative damage to bases leads to the third type of spontaneous mutation, that is active oxygen species, superoxide action, hydrogen peroxide, hydroxyl radical. So these are the different things which occurs here. So produced by the byproduct of the normal aerobic metabolism. So you can see here in this particular scenario that in the environmental sources, that is by smoking, indoor cooking, fires, then airborne pollutions, car exhaust, and what it damages is it actually damages the mitochondria. Also the inflammatory cells like neutrophils, macrophages, epithelials, and endothelial cells, by the xanthine oxidase and the NADPH oxidase, what happens is they will have the ROS system. So the damage is actually taking place in the mitochondria, and also in the other things like tissue damages are there, like lipid peroxidation, and carbonyl stress will take place. There are different scenarios or damages can occur in our body, like mitochondrial damage, autoimmune induction, pro-inflammatory function, then impaired steroid function. So the induced mutation, if you see here, the induced mutation, it leads to the those which arise after the purposeful treatment with mutagens or environmental agents which are known to increase the rate of mutation. Involves incorporation of base analogs, that is specific mispairing by alkylating agents, base damage due to radiation, etc. And there are three agents, main agents, which are discussed here, that is radiation, chemical agents, and transposon insertion. So based on this radiation, if you see, they considered as X-rays, gamma rays, neutron bombardments, and induced mutations. That is the induced, what the mutation is, point mutation. So what happens is if a regular exposure to the X-rays or gamma rays is there, the point mutation can have change in single nucleotide or deletion in chromosomes. So ionizing radiation, if you see, they have the results in the ionized or the excited molecules in the normal level to the higher level, and damages the cellular component or DNA, results in hydroxyl radicals, superoxide radicals, hydrogen peroxides, then damages the bases, causes different proteins or products, causes apurinic site. As we have seen during the replication, if the apurinic site is there, that site will be empty. So the pairing will not take place properly and the mutation can occur. And also the strand damage or strand breaking, and lethal effects in the DNA. So which will be seen in the few years to few, almost like a 10 years, 15 years period, it can be seen. Chemical mutagens, if you see, it causes point mutation, change in single base pair, that is causes or occurs due to transition and transversion mutation. And types which include in this is basically the alkylating agent, base analogs, and intercalating agents like ethidium bromide and others. So alkylating agents, so base analogs and intercalating agents like ethyl methane sulfonate EMS, ethyl ethane sulfonate EES, and mustard gas. So the causes effects on both the replicating and non-replicating DNA. So base analogs, if you see 5-bromouracil, 2-aminopurine, 5-iodouracil is also present here, similar in structure with purines and pyrimidines, and they mutate the DNA at the time of replication. So if you see here, the thiamine, if it is getting mutated, that becomes as 5-bromouracil or 5, that is the keto form and enol form. So during the replication, what happens is this 5-bromouracil, they can actually pair with adenine. But in this adenine pairing, we know that they can also initiate the pairing of third bond also. That is with guanine, if you see, they can pair with guanine also. So what happens is the keto form of 5-bromouracil, they can pair with also they can pair with bromine, guanine. So the triple bondage formation can occur as well as a double bondage formation will occur with adenine and guanine here. So due to this, what happens is there are so many changes which occurs in our system that replication can be altered. That is a particular gene expression can be altered. The transcription as well as translation process can have the effect of this particular damage. So intercalating agents basically causing the addition or deletion of the bases during the replication. Example, ethidium bromide, proflavin, then acridine orange, and ICR compounds. So this, what it causes is causes the transition, transversion, and frame shift mutation here. So if you see how it is getting changed, so this is our bDNA before intercalation. Once the ethidium bromide, acridine orange, or actinomycin D, if it is getting added up, you can see that there is a conformational change in the structure as well as the strand itself. So what happens is due to this intercalation, the whole pattern of the strand and the morphology of the particular DNA helic formation actually changes, due to which sometimes the cell can undergo the death also. So the sudden death can also occur, or sometimes the mutation can also occur, which can be very deadly in the future. So transposable elements, basically the transposable elements are actually also called as jumping genes. Changes their position from one place to another, leads to the disruption of function in normal gene. This is now used by many scientists to identify the new mutation, and this approach is called as insertion mutation. So you can see here, these are the gene 1, 2, 3, and 4 are there. Excuse me. So these are actually the, if you see the transposable elements in this, what happens is if the transposable elements like containing the gene 1, 2, 3, 4 are getting inserted in the normal DNA strand, then what happens is, or in the chromosome, then what happens is the size of the chromosome actually increases. So when the size of the chromosome actually increases, the factors can also change. That is the replication, everything process can also change and can give a new feature during the process. So I hope that in this video you have got an idea that how the process of spontaneous mutation and how the process of induced mutation are taking place in our body. Hope to see you in the next videos with more detail about prokaryotic gene transcription and translation and eukaryotic gene transcription and translation. Thank you and have a good day. Bye.